Yes, it's Uncle Jegz. Last time we looked at vaccination and the difference between an explanation and an evaluation question. Click the card to teleport over to that video. This video, we're going to look over antibiotics and painkillers. We're going to see how they're used, and also we're going to look at bacterial resistance to antibiotics. Well then, let's chop some science. Here are the retrieval questions for this lesson. You know what to do. Vaccines contain weakened or dead pathogens. Measles is caused by a virus. 55% of blood is plasma, 45% are red blood cells, and less than 1% are your white blood cells and platelets. You may notice that this adds up to more than 100%, but that's just due to the way that we've rounded these numbers up. Funny thing is that I made this lesson a while ago, but due to recent developments around the world, this idea has become a trending topic right now. Spend a few seconds thinking about this. Bleach is very good at killing pathogens, so why don't we inject it into people that are infected? The issue with this is that bleach doesn't discriminate. It will finish the pathogen, but it will also ruin the homeostatic party inside your body as well.
Oh man, you're just ruining it. Look at my lips, you're ruining it.
Chemicals like bleach are way too poisonous to use inside your body. They kill you along with the pathogen as well. I think we ought to clarify the differences between these substances. Antiseptics, antibiotics, and antibodies. Match the substances to their correct boxes. Antiseptics like hand sanitizers kill microbes outside of your body, like on your skin or on the kitchen counter. Antibiotics kill bacteria inside your body, but they don't kill viruses. Antibodies, on the other hand, as we saw in the last few videos, kill both bacteria and viruses inside your body. Here's a little challenge activity for you. Jaden,
Wagwan.
Where would you categorize painkillers? Antiseptics, antibiotics, or antibodies? You guys at home can also have a go. Well, if you said antiseptic, you're wrong. And if you said antibodies, you're also wrong. But if you said antibiotics, you were wrong as well.
He lied!
Yeah, sorry about that. Painkillers are drugs that reduce the symptoms of an illness. Let's say a pathogen gets inside your body and starts damaging some of your tissue. Your pain receptors send pain signals to your brain, which makes you feel the sensation of pain. One of the actions of painkillers is to reduce the chances of your pain receptors firing. This reduces the sensation of pain that you feel. The pathogen is still running amok inside your body, but you're just numb to it. Your receptors are just ignoring the call, like. Here's another challenge question for you now. Which of these would a painkiller treat? Headache, sore throat, or infection? Have a go at this. Painkillers will treat both headaches and sore throat because these are both symptoms of an illness. They cannot treat infections because they don't kill viruses or bacteria. Remember, this is how viruses work. They attach to your cells, the surface proteins in your cells, and then they inject the DNA or RNA inside your body. Once the DNA or RNA is inside the body, in this example we're using a RNA type of virus, it changes its RNA into DNA using an enzyme called reverse transcriptase. Then when that DNA is fully formed, you can see it's a double strand now, It can go inside your nucleus and integrate itself into your genome. We saw in a few videos ago that roughly 8% of your DNA is actually viral DNA. This is why it's difficult to kill viruses inside your body. Drugs that kill the virus are likely also going to kill your body cells. This is because the virus is inside the host cell. It's not lingering outside, it's actually inside your cell. And so drugs that kill the virus are likely to kill your body cells as well. But if you have a careful look at the GIF, you will probably be able to spot how your body cell can probably discriminate between your normal uninfected body cells and other cells that have been infected. Let's quickly look at how penicillin was discovered. As the story goes, this breeder, Sir Alexander Fleming, was studying a bacteria in a petri dish and couldn't be bothered to clean his equipment. He left the dirty dishes in the lab over the weekend. When scientists grow bacteria, they use something called agar plates. Agar is a jelly-like substance that's used in desserts in places like Japan. Trilogy students do not need to know this, but if you want to grow your own bacteria, this is what you have to do. You pour hot agar gel into your petri dish. This gel will also have a mixture of your nutrients that the bacteria is going to need to grow as well. You leave the gel to cool and solidify. You sterilize the dish to kill unwanted microbes. You then inoculate the agar plate with your microbe. Then you incubate for a few days and watch the bacterial colonies grow.
I want you to watch over me. Hey! Wow! 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 That is how your life should be.
And as you can see here, some lovely colonies of bacteria started growing just like what we saw in Fleming's photo. The GIF on the right wasn't long enough to show you that the petri dish was going to be littered with these bacterial colonies. Now it's time for us to look at why this one picture right here, this one picture here changed the course of human history. It shall go down as one of the most important things that's ever happened in human history.
Never, never, never forget.
I've got some challenge questions coming up, but you need to deep these two pictures first. Look closely at what's happening on the picture on the left. Now look closely at what's happening on the picture on the right. Here are the challenge questions. The first one gives you a sentence of information and then asks you to state what's happening in the top image. The second one is asking you to describe how penicillin works, and this is 2 marks. The third one is asking you to state why penicillin doesn't attack human cells. This is another 2 marks. Pause and have a go. In the top image, we see this white stuff. It's a fungus called penicillin. Notice how there's bare colonies on the other side of the petri dish. Also notice how the colonies are smaller the closer you get to the fungus. Fleming noticed that the fungus was killing or inhibiting the growth of the bacteria. He deduced that the fungus must be producing something. The bottom left picture shows that the penicillin is rupturing the bacterial cell wall, which would obviously kill the bacterial cell. The last picture at the bottom primed you to remember the differences between bacteria cells and human cells. We saw in the previous question that penicillin attacks the cell wall. Animal cells do not contain cell walls, and so the penicillin cannot affect it. If you need to refresh your memory on prokaryotic and eukaryotic cells, clickety-click this card over here to watch that.
And what kind of grammar is that?
Business grammar.
Alright, so antibiotics kill bacterial pathogens. Here's a graph showing the death rate of syphilis and AIDS. Here are some challenge questions for you to have a go at. You know what to do? For 3 marks, you need to describe the trend after 1940. Describes means state what the graph is showing. we see that the deaths from syphilis have decreased, but that after 1980, the deaths from AIDS has increased. When you have to explain the graph, you have to give reasons why the trend is the way it is. Penicillin can treat bacterial diseases like syphilis, but it can't treat viral diseases like HIV, which causes AIDS. Also, HIV didn't start until 1980s. Up until this point, a bacterial infection could easily have been a death sentence, but due to Fleming's messiness, bacteria were done out here.
I am never going to financially recover from this.
And then, after taking that major L, natural selection came over and blessed these pathogens. The pathogens were about to be eliminated, but then they just said,
Impossible. If they try it, I will launch a counter-attack wherever I am to teach them the lesson of their lives.
We have covered these two parts of the specification. Now we're going to look at what happens when we get carried away with antibiotics like penicillin. Actually, before I show you, try and match up the drugs that are antibiotics and the ones that are painkillers. Aspirin is a painkiller. Penicillin is an antibiotic. Paracetamol is a painkiller. And amoxicillin is an antibiotic. Notice that the two antibiotics end in "-cillin". That is because amoxicillin is named after amino, oxygen, and penicillin, which will give you an idea of what the structure is probably like. But not all antibiotics are similar to penicillin. We can categorize antibiotics into two groups: broad spectrum antibiotics and narrow spectrum antibiotics. Broad spectrum antibiotics can kill a large range of bacteria, but narrow spectrum only kills a specific group of bacteria. It is very important for your doctor to prescribe the right one for you, because this has contributed to bacterial resistance. To understand bacterial resistance, you first need to deep evolution by natural selection. The first step is that a mutation occurs in your DNA. A mutation is a change in your DNA. And because your DNA codes for your characteristics, what you look like and some parts of how you behave, a change in your DNA will change your characteristics. And differences in characteristics are called variation. Variations in humans are stuff like hair color, eye color, things about us that are different. Some variations are better suited to the environment than others. In this picture, we see the same species with different variations. In this environment, the lighter moths are better adapted and so don't get munched as much as the darker ones. The successful organism, the lighty moths, then survive long enough to pass on their useful genes, the genes that made them light. This then increases the variation in that environment. That's because the darker moths are more likely to be eaten than the lighter moths, so there'll be more lighter moths because they're passing on that useful gene. The key point to stress here is that the environment is what's selecting who lives and who dies, natural selection. The fact that the environment was lighter meant that the lighties were better adapted to pass on their genes, to live long enough to pass on their genes. There is no purpose to this. Natural selection is just like gravity, it's natural, it just happens. If the environment changed now, then the selection pressure changes. Pre this. When soots from factories after the Industrial Revolution started coating the barks of trees, the selection pressure favored the darker moths. Now the darkies were the ones that were able to survive long enough and also reproduced more, and then the population of darker moths increased.
I think these lighties loving my black gang. Everyone suits same colour as Batman's.
Let's now look at how to apply this to bacteria. MRSA is a resistant form of a common bacteria. The M starts for methicillin. You can work out what that must be from the -cillin ending. R is resistant, and the SA is the genus and the species of the bacteria. So Staphylococcus aureus.
Jesus Christ.
The name looks a bit crazy, but it all makes sense when you understand a little bit of Greek and Latin. Staphyli means a bunch of grapes in Greek, and coccus means berries. Look at this image, and then you can probably work out why they chose to name that genus Staphylococcus. It looks like a bunch of grapes. And then the end part, the species name aureus, is due to the fact that the colonies of the Staphylococcus aureus looked a bit goldish. And if you have a look at your periodic table, you notice that gold has the letters AU. That's because it comes from the Latin word for gold. So here are the steps in bacterial resistance. There are 4 main steps. First of all, just like before, a random mutation creates a resistant bacteria. The patient takes antibiotics and then the antibiotics kill the non-resistant bacteria. This is the selection pressure from the environment. The surviving bacteria all are likely to be resistant, and so they reproduce because they have little competition from the non-resistant bacteria. They've all been murked. This then makes it more likely that the next generation has a higher level of resistance. So now we have a situation where more and more bacteria are getting a little bit too brave.
Do me a favor, please. Get out of here. Get out of here, man.
And would you look at that? Lesson done.
Fatality.
We looked at the differences between painkillers, antibiotics, and antibodies. We then looked at the discovery of antibiotics and also bacterial resistance. Check the description for the timestamps and other goodies. Hope you found all of that useful, and I'll catch you guys in a bit.